Thanks everybody for coming out on this rainy Tuesday. We're practically like submerged within the ocean today. I felt like driving through my neighborhood, definitely at sea level here. So today I'm going to talk to you about some of the work that CCMI has been doing over a long time period and really how important those kinds of data sets are for understanding change over, what is happening on our reef, how can we predict what it's going to be like in the future, and what can we do and what we are doing at CCMI to try to promote conservation or just the resilience of the coral reef that we have here. So I'm going to start with a bit of background. If you've been to one of my lectures before, you've probably seen this slide already. This basically is talking about how in the Caribbean and really globally there has been a continuous decline in coral reef health and the cover of coral on our reefs systems for several decades, and that really started in the mid-1970s. So around that time frame, coral cover in the Caribbean was somewhere around 70%. And then there was a series of events that led to their decline. One of the first was a hurricane, followed by disease outbreaks, and then coral bleaching. And this has led to an overall percent cover hovering around 5 to 10% now for the Caribbean region-wide. Now, this is extremely disheartening and sad, and especially, for people like Jeremy Jackson or Terry Hughes, who were in Jamaica in the 1970s, and they were taking pictures like this, of these huge fields of AGRRA, to then go back now and see mostly algae. You can only imagine how heartbreaking that must be. Interestingly, young people coming to work with us, they go out onto the reef and they think it's amazing. They're like, this is so great. And so I think that's a really important thing to remember, is this concept of shifting baselines and what is your perception of what a healthy reef is. And that also harkens back to how important it is that we have these really long-term data sets so we can say, yeah, it looks okay, but it used to be like this. So we have something to reference. And there are a lot of reasons why this decline has happened, and it's been discussed in the coral reef scientific literature for several decades. You can see that this is the percent degradation on the y-axis, and so you can see that the dots represent different regions. So we've got Cayman Islands and Bermuda here, and you can see that that percentage of degradation on the reefs really started happening in the mid to late 1900s, so 1970, 1980 time frame. Now, we think that this has a lot to do with industrialization, industrialized fishing, as well as the contribution of pollutants into the ocean environment. And this led to things like increases in diseases, excessive algal growth, and some were promoting this idea or hypothesizing that we were on what they were calling a slippery slope to slime. So this is the graphic that they talked about in this paper, where you have this continued degradation and then it becomes more and more difficult for your reef to recover back to that pristine state. And we've seen that remarkably in the Caribbean with our AGRRA corals, our staghorns and our elkhorns. So in the early 1980s is when we really started seeing this complete loss of the staghorn corals, and this was related to a coral disease, white band disease, that spread throughout the Caribbean and killed over 90% of those corals. White band looks similar to this, but there are many other diseases that impact corals and have caused degradation. Things like black band disease, this is white plague disease, yellow blotch disease, as well as coral bleaching. And coral bleaching is primarily caused by an increase in seawater temperature. So corals live pretty much at the upper thermal limit that they can tolerate. So even an increase in seawater temperature of just 1 degree Celsius can result in corals bleaching and eventually potential death for the coral. And because of that, we know that global temperatures are rising and global seawater temperatures are rising. And this has resulted in global bleaching events that are occurring more frequently now, almost annually, as we're moving into 2020, 2021. But we can only really figure out why these shifts are happening by looking at long-term data sets. So it's really important to be able to look back in time and try to correlate these losses with changes in the physical environment, changes in the fish abundances, or changes in our practices that could be instigating this decline. So there are two such programs that are looking at changes in the physical environment and have been doing so for a very long time. So one is in Hawaii, it's called HOTS, the Hawaii Ocean Time Series, and the other is in Bermuda, the Bermuda Atlantic Time Series. Both of these programs are ocean-based, so they go out on large ships and they're measuring properties of the ocean like temperature, light, the amount of phytoplankton in the water, the amount of the chemical composition of the water, and we look at that over time. So for example, the amount of carbon dioxide in the water. And it's actually these data sets from these two programs which were the first to really pinpoint that the ocean was in fact warming and the rate at which ocean warming was occurring. So again, this access to these kinds of long-term monitoring data sets are so important for the science and really figuring out what's happening on our planet. You know, it's not necessarily the sexiest science because nobody really likes to pay for monitoring programs, but it's a necessary thing for us to do in order to be able to actually make any real good predictions. So here at CCMI, we have the goal of assessing coral reef health in the Cayman Islands, and we've been doing this for quite a long time. And so this is done by doing a series of surveys around the island of Little Cayman. And so we're looking for changes in the composition of the reef, so both in the corals and in the fish. So this is our lab here. If you haven't been out to Little Cayman, when you do, we encourage you to come stop by. There's always people around the station, and we're happy to give you a tour and show you around. But we are really lucky because we're right in front of the reef. You know, this beautiful, what would be considered pristine for the Caribbean reef is just there right at our fingertips. So every year since 1999, we have been going out with a team of researchers or whoever we can find really who's working at CCMI at the time. It can be a team of interns or visiting students. We train them to do coral ID and fish ID, and we go out and do a survey every year in between July and August. And this is using a standardized protocol by the Atlantic and Gulf Rapid Reef Assessment, or AGRRA. And these data then provide a baseline for change. So we can then look back every year since 1999 and see how much coral there is, how many fish there are, what types of coral there are, what types of fish there are. And this isn't going to allow us to provide that really critical long-term data set to give to managers like the DOE and help them discern our marine protected areas working, for example. How can we change our management strategy in order to protect what we have left? So during these surveys, we go to 6 to 12 sites every year. We aim to do at least 10 sites, really. And so on each dive, we're doing a survey for corals. So we identify, count, and measure every coral that we encounter along a transect as we're swimming. And then we assess its health. So is there anything wrong with it? Is it bleached? Does it have a disease? And then we also take a quadrat down, which is basically just a small square that we place on the bottom, and then we assess the percentage of algae, sand, coral, sponges, etc. in the square. And so that will give us an idea of what the benthic composition is. And then we have a second team that is also doing counts of fish. So they swim along a transect line and count and estimate the size of every fish that they see along a 10-meter transect line. All right, so without further ado, what we have found. So this is our percent coral cover over the last 22 years. What I find really encouraging is that we are at a high point here in 2020. So we are still maintaining roughly 24% coral cover, which is among the highest in the Caribbean. So that is something for us to be really proud of. And I think it really shows that the protections that we have in place here are working. One thing I think is interesting about the data, however, is this cyclical pattern that we see. And that, again, is something that you would never be able to pull out in, if you were just going 1 or 2 years in a row. So I'm interested to try to dig deeper into our data set and figure out why we're finding these peaks and troughs in coral cover over time. I think the positive thing is that we're back up. So we're on an uptick in coral cover. And, in the face of things like stony coral tissue loss disease, I'm hopeful that Little Cayman may in fact be more resilient than other locations because perhaps it has a higher biodiversity and a higher inherent resistance to that disease because of this high coral cover, but that waits to be seen. Interestingly, though, we have seen a shift in the species of corals that are present on the reef since 1999. So in 99, we had the majority of the corals were orbicelids, which is this coral here, which is a star coral. So for example, if you're diving Eden Rock, they're the large, big boulder corals. A lot of them almost look like clusters of ice cream cones with a coral on the top of it, is what I kind of think of them. That's the orbicella. So that was the majority of the coral that we were seeing on our transects. The very top little sliver is the AGRRA. So there was still some AGRRA in our surveys in 99, but that has since disappeared. What we did find was an increase in the porites, which are small little colonies. They're usually like a mustard yellow coral, or sometimes they're fingering small branching corals. And the agariciids, which are lettuce-like corals, and they are really all over the reef now. So those are the dominant species. And this is important because this is a shift not just in from one species to another, but the kinds of corals that these are very different. So the orbicelids are huge boulder corals. They will form these massive colonies. They're very important in keeping up our calcium carbonate structure of the reef. Porites and agaricea are generally small colonies. They don't grow very big. They're much less important in terms of reef builders. And so they don't add to the complexity of the topography as much as those orbicelids. So this could result in a shift in the entire community, not just the corals, but this can also then affect things like the fish community that live in this reef structure, or even things like lobster, if there's not as many places to hide, for example. But what is hopeful is that we have seen a continuous decline in algal cover. So this is looking at those quadrats when we look at the square and we assess how much algae is in every square. We have in fact seen a significant decline in algae over time. And this is really great news for the reef, and it indicates that potentially the herbivorous fish population is thriving in Little Cayman. And in fact, we have found a significant increase in fish over time. So this is the density of fish. So the number of fish that we're counting along our transect has actually increased since 99. And what I found really encouraging, because every time we do these surveys every year, the group of people doing the surveys is slightly different, and you never really know, how good each individual is at counting the fish or measuring or IDing the fish. But when I saw that our number of fish was almost exactly the same as last year, I was like, okay, our team got it right this year. I'm pretty confident in that. If we had just had this crazy spike out of nowhere, I would have been a little concerned, but we don't. So I think it's positive. I think it means that there is a high density of fish in Little Cayman, and if you dive there, I think you would agree that there are a lot of fish. And so going back to that algal cover, what's important is, what kinds of fish are these that are living on our reef? And in fact, we have found also a significant increase in parrotfish over time since 1999. So parrotfish are probably the most important herbivore on the reef. They eat a lot of algae. They scrape off the turf algae, but even that really foliose algae that's growing on the reef. And they keep the reef clean and enable corals to recruit and grow and survive. So having an increase in parrotfish is probably one of the main reasons that we're seeing this continued recruitment cycle of corals on our reefs in Little Cayman and sustaining that high % coral cover. If we look at all the different types of trophic groups of fish, so here we're looking at carnivores on the top, corallivores, herbivores, and omnivores. And the only actual major trend we saw was an increase in herbivores, which is this section here. So this was in fact a significant increase. Everyone else kind of stayed the same. So while we are finding this increase in herbivores, we haven't seen that transpire up the food chain yet. The hope is that as we continue to protect our fish in Little Cayman, that eventually as the population continues to rebound, we will start seeing an increase in those carnivorous species. So probably, in a few years' time, we will actually start seeing more grouper on our survey and more sharks. Because it does take a little bit of time. There's a little bit of lag time. And especially now that the Cayman Islands has increased the size of the marine protected area in Little Cayman, I'm very hopeful that we will see that shift in the other trophic groups as well in the future. And why is this important that we have all of these different trophic groups? Well, there's been a lot of work that has indicated that having a diverse fish population actually leads to increased coral health. So it's not just having a healthy coral reef helps the fish also help the coral. And so we need both of those to be healthy and thriving in order for the entire ecosystem to work. So this study was done in the Line Islands in the Pacific. And they were looking at a series of islands that had, so starting from the bottom was an inhabited and dense population island up to an uninhabited island. And what they found was a direct shift in the fish composition as well as the percent cover of the corals. So the highest percent cover of corals was found in sites at that uninhabited island, and there were a lot of sharks, a lot of major carnivores present. And as you moved down and there was more and more human impact, you started to lose those top trophic groups, and you end up with small little, omnivorous little invertivores and no more carnivores. And eventually no more herbivores, and then you're looking at just algae. So it's really important that we do continue to protect the fish in the Cayman Islands, because this will inherently then protect the coral. And we are finding really positive signs. So one of the things with the Healthy Reef report card is that we assess what we call the state of the reef. And so this is based on just the percent cover of each reef that we're surveying. So of those 10 sites, how many had less than 5% coral, 5 to 10% coral, 10 to 15, 15 to 25, and 25 and above, right? And so what we ended up finding, the highest group is very good. On here, we don't even have critical because we didn't have anything that was less than 5% coral cover. But we did find, you'll see again, this cyclical pattern is kind of present here because we're looking now every 5 years. So 99, 2005, 2010, 2015, 2020. And you can see again this, the reef is really healthy. It's not so great. It's really healthy. It's not so great. It's really healthy. So we're in this really healthy stage of the reef. And in fact, it's actually based on this report card metric healthier now than it was in 1999 if you were just looking at the percentage of reefs that fell in this higher than 30% coral cover group or the very good group. So this is really positive news. It means that the corals are in fact doing really well, and they are taking up the majority of that reef, that benthos. So how will they fare? So we've looked at how they've done over time. And the next thing we want to do is look at, okay, how are they going to perform in the future? And some of the major things that we've talked about tonight that can impact the health of the coral reef are things like exploitation, overexploitation, but also this increase in seawater temperature associated with global climate change. So one of the ways that we are trying to assess how corals will survive in the future is through experiments in the lab. And so we're looking at the tolerance of different individual corals to deal with temperature. To do this, we're creating these what we call thermal performance curves. So this is an example of a thermal performance curve. And you can think of the performance as any really metric of biology. It could be growth, It could be reproduction. If for our purposes we use oxygen metabolism, or how much oxygen the organism is consuming over time under that temperature. So when we were thinking about, can corals survive increases in temperatures, one question that comes to mind is, well, is it dependent on where they live and the history of that environment? So for example, if I grew up in a very hot climate, does that mean I'm more tolerant of hot temperatures? Or if I grew up in, northern Canada, does that mean I am less tolerant of very high temperatures? And so this was sort of the initial question that we had. So thinking that if the coral lived in a very warm environment, its thermal tolerance, or the highest temperature it could tolerate, would be shifted towards warmer temperatures compared to colder areas. I'm going to skip that one. So we did this in 2 different locations that we considered hot and cold for coral reefs to be growing in. Bermuda being the cold zone. So in this top window here, you can see the temperature of the seawater. In the blue is Bermuda, so it dips down in the winter to about 16, 17 degrees C. And in the summer, it peaks at 30 degrees C, whereas in Panama, it hovers around 28 to 32 all year long. And so we were looking at, does this performance or the ability to tolerate temperature differ between the same species in these 2 different locations? So we go out onto the reef and we collect corals, we bring them back into the lab, and we actually use a diamond-edged saw and cut them into tiny little squares, so they're all roughly the same size and can fit in these tiny little chambers. And these chambers then have a temperature probe and an oxygen probe, and we can adjust the temperature of the water bath that it's sitting in. And so we can look at then how they're performing over time in a given temperature. So we did this for 10 different temperatures, and we measured their oxygen metabolism using this very fancy machine. And what we found was, in fact, what we hypothesized. So the corals from Panama, which are this red curve, in fact had about almost 1 degree higher thermal tolerance than the corals from Bermuda, which means that they did in fact learn or inherit, or there's the history matters. Like what they had dealt with in the past has adjusted their ability to tolerate temperature in the future. This I think is very hopeful news, because it means that perhaps corals, as they are experiencing heat waves or continuous exposure to heat, they may be able to then shift their tolerance to higher and higher temperatures with time. It's just, is that going to be fast enough to deal with the increase in water temperature that's occurring on the reef? So the next question we had was, how does this relate to depth? And do corals that are living at depth then also have this sort of different threshold for temperature than corals that live in the shallow, because temperature is buffered with depth? So we did pretty much the same experiment, but just along a depth gradient. So we collected corals from very shallow, about 10 meters deep, and then collected them from, I think this one was 35 meters deep. And we did the same experiments looking at their oxygen metabolism. So you can see what I want you to look at is the difference between the light and the dark lines, which directly overlap. So that's the difference between shallow and deep for respiration and photosynthesis. And there was no difference. So this means that depth, in fact, was not affecting their tolerance. So they were not so adapted to the local conditions at depth that they couldn't tolerate a spike in temperature. This is great news. Could potentially mean that deep reefs could, in fact, serve as a refuge for corals to survive as seawater's temperatures continue to rise. So how does that relate to us here at CCMI and what we want to do for our corals and how we can protect our coral reefs? Well, you may know that we have a very large coral nursery, and we are growing acroporids in our nursery that with the intention to outplant those corals out onto the reef and restore the shallow reef to something resembling what they used to be in the 1970s. We will never get to that point, but at least we're trying to keep that species alive and established within the population. So what we're trying to do now is use this method that I've done in the past with other corals, with these staghorn corals in the lab. So we'll go out to our nursery, collect several corals from the nursery, different genotypes, different individuals of the same genotype, just a broad range, bring them into the lab and assess their thermal tolerance or their capacity to tolerate heat. And then we can potentially select individuals which we think are going to survive higher temperatures and outplant them onto the reef to create a new reef that is more resilient to global climate change. So that is ultimately what our goal is, and what we are hopeful that we, by doing these kinds of experiments, promoting the genetic diversity at the same time within those populations, that we will in fact be building more resilient coral reefs. And with that, I just want to thank everybody for coming and listening. I know it's a lot to listen to after work on a Tuesday evening.